Thank you very much. I appreciate the invitation. It's wonderful to be back in person this time. During COVID, the interactions left a lot to be desired through a screen, so I'm delighted to be here. Thank you for the kind invitation. We'll talk about bioelectronic medicine. I will explain what that is and technology that we're developing to help it propagate to patients at scale. So we're at the onset of a new era where disease is being treated by electronic devices that interact with the body in a bidirectional fashion. So these devices are typically implanted inside the body. They can measure the condition of the body. They latch onto the nervous system. They can measure the signaling there and then stimulate the nervous system to help the body go back to homeostasis. This is a massive departure from the current paradigm where pharmaceuticals are interacting with the body one way, where you would release it into the bloodstream and hope that it goes in the right place and has the right effect and it doesn't have any side effects. A quick declaration of interest. I'm involved in a couple of companies, and I'll show you some data that are on the commercial sphere. So it's up to you to believe me or not. So this is bioelectronic medicine, one example of a technique called deep brain stimulation. So here is a patient who has tremor. This is a neurological, it's a movement condition that is manifested by this uncontrollable motion of the arms that makes daily life difficult to live. And with this intervention, whereby an electrode array is placed in the subthalamic nucleus, deep in the brain, and tickles that structure with electrical current, you have an immediate and impressive amelioration of the symptoms. So with a flick of a switch. The history of this intervention of the whole field of bioelectronic medicine heralds back to the ancient world where electric fish was used to deliver treatment to people with migraines or gout. The efficacy and safety of this technique is not very well documented, but it was in the 18th century that Galvani started putting the field on a scientific basis. Famous experiments with a dead frog, applying electricity, getting the legs to twitch, gave rise to a lot of speculation about the nature of the nervous system. And 200 years later, there were already medical devices. This is a cardiac pacemaker. The year before these devices became fully implanted. So at that time, the patient had to carry with them this life-saving card of batteries and electronics. In 1958, there was the first fully implanted pacemaker at the Karolinska Hospital in Sweden. And that kick-started the modern era of bioelectronic medicine. So here the intervention is on the heart. Electrodes placed on the heart can pace the heart to help people with arrhythmias. Then a decade later, late 60s, cochlear implants were developed for people with profound deafness that cannot be helped with external devices. The intervention here is on the in the cochlea, the hearing organ next to the brain. Then spinal cord stimulation for helping people with chronic pain, a couple of other of peripheral nerve prosthesis, until the brain came to the forefront in 2000 with deep brain stimulation for neurological and neuropsychiatric disorders. And then the floodgates opened up. And many, many devices were approved for use by the FDA. This is not a comprehensive list, but includes a couple of interesting devices. For example, peripheral nerve stimulation, stimulation of the vagus nerve, is used today to expand the target of indications that can be treated to autoimmune diseases, including rheumatoid arthritis, type 1 diabetes, and others are believed to be treatable with this technology. What are the limitations? This sounds great, right? You put a little implant, tickles the nervous system, and you cure disease. Why don't we see this deployed to patients at scale? There are some formidable challenges. First and foremost, we do not understand how the brain works. It's an exceptionally complex system in human, consisting of over 86 billion neurons connected with as many as 1,000 connections per neuron. So in numbers, it's astronomical. We do understand a lot about how neurons communicate with each other at a cellular level. We know that neurons are electrically active cells with processes that are called dendrites. They collect information. That information gets compiled, and then an action potential, which is like a voltage pulse, comes down this long process that's called the axon. And when it arrives at the terminus here, the synapse, it leads to the secretion of neurotransmitter that can either excite or inhibit the postsynaptic neuron. So what I just narrated is understood in a quantitative basis. However, what is not understood is how do you go from this cellular phenomena to behavior. In order to close that gap, electrophysiologists are using electrodes that can be either implanted into the brain, and if they're small enough, they can measure single neuronal activities. Cortical electrodes that are placed on the surface of the brain and typically look at networks of neurons. And then scalp electrodes, where this technique is called electroencephalography, where they can look at network type of activity. And by combining these techniques, hopefully you will one day be able to solve this puzzle. How do you go from neuronal activity to behavior? The opposite is also not understood. Why, when we stimulate the brain with electric current, why do we change behavior? What's the mechanism of neuromodulation? Now, these fundamental limitations go hand in hand with current technological limitations. For example, the manufacturing is not scalable, and this prohibits the technology from being deployed. And that limits the knowledge that we collect back from these devices. It's highly invasive. Typically, you require invasive surgery to place these devices. And poor specificity is something that current devices unfortunately suffer from because they have rather large electrodes. They tend to stimulate areas that shouldn't be stimulated. So there is a lot of scope for new technologies to address these limitations and enable bioelectronic medicine at scale. This is a snapshot of the present different devices that are available, placed at different locations, with the indications that they treat here in red. And by and large, all devices look like this. There is a tin can that contains about 2/3 of the volume, the battery, and 1/3 electronics that create current pulses that are taken through these leads to electrode arrays that are placed in the location where intervention is needed. This is the case for deep brain stimulation, 2 electrode arrays, one in each hemisphere. And this can is placed under the skin here on the chest. So once placed, electrodes are supposed to stay in for the life of the patient. This little can comes out every decade or so to change batteries. These devices are made by hand. In the year 2025, if you go and visit Medtronic, Boston Scientific, Medel, all the big companies, that's how their fabs look like. So you have people working through microscopes with tweezers, putting little electrodes together. This is incredible. This is not scalable technology. It heralds back to mechanical watchmaking. And the reason why it has persisted for so long is because of the extreme reticence of this community. Microelectronics is manufactured in a completely different fashion. So this is TSMC, the biggest microelectronics fab in the world. And you see very few people. Here, machines feed wafers into each other. And they do processes such as film deposition from atoms, lithography, etching, and out of that come objects of exceptional complexity, precision, reproducibility at an ever-decreasing cost per functional unit. So this technology is slowly entering now bioelectronic medicine, and it's going to change it quite dramatically. Now, I have a couple of slides with open skulls and blood, so if you're sensitive, there will be a warning, look away. So what we can do today is go from this to that. What is this? This is exploratory brain mapping before surgery. So here you open the skull, you do a craniotomy, you do a durectomy. This is the membrane that encloses the brain. And then you can see the cortex underneath. And you slide this electrode array in order to map the electrical activity so you know where to resect and where not to. Unfortunately, the resolution is very poor because this device is made by hand. It's millimeters in dimension of electrodes. And that's the clinical standard today. What we can do, though, is go to this situation where through microfabrication, you can have hundreds of contacts here and map the brain with micron scale resolution, getting down to single neuron activity. And that not only will allow more precise diagnosis and help resection, but also will teach us a lot about the brain. This is a similar technology with the one that Elon Musk is running with. Here, this is how their electrodes look like. It's linear arrays, again, of microfabricated electrodes that have the size typical of the soma of a neuron, so of the order of tens of microns. These are implanted into the motor cortex in people who are paralyzed, and they record from the motor cortex volition for movement. And then a computer can translate that into, for example, moving a piece on a chessboard or driving a prosthetic arm. So with that as an introduction, I want to give you a couple of snapshots of work that happens in labs around the world to make technology more deployable at scale. And one example is shape adaptive implants, implants that can change their shape, and how can they help us with some of the problems facing in the clinic, we're facing in the clinic today. One example is in spinal cord implants that are used to treat chronic pain. There are two technologies there. One is this percutaneous, as it's called, array. It's a linear array of electrodes that can be placed on the spinal cord through the space between the vertebrae. So here you would find the space between the vertebrae, jab a needle there, and then take this device through the needle and place it on the spinal cord. This is a lumbar puncture procedure similar to a spinal tap where you remove cerebrospinal fluid, or to the delivery of anesthesia, intrathecal anesthesia. So facile placement is a big plus of this device. Unfortunately, once placed in, it can migrate as the person who wears this device goes about their daily life. That device might fall out of alignment with a part of the spinal cord it should align with and lose efficacy. And then the person has to go back to the clinic for revision surgery. Also, because it's rather small, it addresses a small part of the spinal cord. So the treatment it delivers is not as effective as it could be. The paddle electrode is a much better technology. Once placed on the spinal cord, it overlaps with a much larger area, so it can deliver better treatment. Plus, it doesn't migrate. It stays put. However, to place it in the body, you have to do a laminectomy. You have to remove part of the vertebrae, which requires the hospitalization of the patient. So it's quite a tough choice to have to make as a patient today. Do you want a device that can go in easily but might not work after a while, or you want to undergo a laminectomy and then have an efficient device placed in? Can we get the best of two worlds? So here, Ben Woodington, very talented student in the group, he developed a technology where he took these thin film electrodes that I showed you earlier for placing on the brain, and he equipped them with microfluidic channels. He could roll them up into the shape of a percutaneous array and then expand them by activating the microfluidic to become a paddle electrode and cover a large area. So the idea here is that you make this device, you roll it up, then you can put it in the body with a lumbar puncture. And once it's positioned where it needs to be, then you can actuate the microfluidic and open it up and roll it, covering a larger area. So this is shown here in a human cadaver. This is the injection site, and this is the device being placed in under fluoroscopy that images the expansion of the device in the body, the location and the expansion. And once it's in the right position, then you can actuate it by pumping some saline, and then it will open up. And you can see this through fluoroscopy. There are two X-ray markers above and below the middle of the device that coincide when the device is rolled up. And then they separate as the device is deployed. And then you can go in and do a laminectomy and find out that the device really opens up and hugs the spinal cord very effectively. So this here and this one there are the two X-ray markers. And this is the injection site. So this is also something that can work in the brain. You can make devices that can be collapsed and then go into the brain through a burr hole. And then once in, they can be pneumatically actuated to open up and cover a larger area. That hopefully will help advance the implantation of these devices by making the surgery much more scalable than currently is. Another way to change the shape of these devices is to use a phenomenon which is called artificial muscles. This goes back to 70s and 80s, but it's coming back today under the umbrella of soft robotics. And the idea is that you have a bilayer structure where you have a thin metal film, and on top of that, you have a layer of a polymer that can uptake ions from a solution and change its dimensions. For example, it can swell. So if you apply a potential between this electrode here and the electrolyte, you can get ions into the polymer will swell and will push the bilayer structure to curl in one way, or when you pull the ions out by applying the opposite potential, it goes the other way. So these devices can do very impressive acrobatics, although the force that they apply is minute. And that can be an advantage if you want to make contact, temporary contact, to a part of the body, for example, for intraoperative monitoring. This is the work of Chao Chun Dong, who's now on her way to Columbia University in the US. So turns out you can tune the structure of this device. If you structure the polymer layer in fingers and pattern them at an angle with the line of the device, then you can achieve other forms of motion. For example, you can make the device do a helicoidal type of motion, as it's shown here, and wrap around a nerve. So we make devices where they are rolled up as fabricated. Then we can open them up by applying a potential, present them to a nerve, stop applying a potential, and then they will wrap around the nerve and hold it gently. If we equip them with microelectrodes, then they can monitor traffic that goes up and down that nerve. There is a need for these devices in intraoperative recordings. When a surgeon operates in an area that has a lot of nerves, they need to monitor nerve health. This is typically done with a device that looks a little bit like a chopstick, where you touch it on the nerve, but nerves being so sensitive, that device can cause damage. So this is an alternative technology that is very gentle. By putting the device on the nerve, you do not elicit any activity. However, if you gently press the paw of the animal, this is on the sciatic nerve, it can pick up the sensory afferents as they go up the nerve. So where are we going with this technology? We're exploring different configurations. For example, this is an in vitro device where you give it an organoid, and you can squeeze it very gently, measure its electrical activity, and then open up to let it develop without any constraints, and then you can measure it again and again. My dream is to make implants that can self-implant and self-position inside the body. There are two needs for that. One is the need to follow tracks that are not linear. You can put the implant in and have it go through a complex track that ensures minimal invasiveness to vital structures. Or you can have implants that through the life of the patient move to explore different areas and see if they can deliver more effective stimulation in the vicinity of where they were first implanted. Now, let's assume that this implant's working. You can wrap around nerves, you can wrap around spinal cord. What could you do? Here's one example of what you can do. Peripheral nerve system, the peripheral nerve system is, receives a lot of attention for neuromodulation. And there are a couple of different implants there. The most common one is this epineural implant, where you have a device that is elastic. You open it up, you present it to a nerve, and then you let it go, and it will curl and capture the nerve and measure the traffic along that nerve. Unfortunately, those devices have a rather poor lifetime because they tend to squeeze the nerve, and they don't fare very well in humans. Their translation has not been very successful. So Alejandro Carnicer developed very gentle implants that can latch around a nerve, causing minimal invasiveness, having minimal invasiveness. And he's exploring the limits of this technology. So here's one example of what you can do with that. Here he's latching onto the three nerves that make the branchial plexus. These are the ones that control hand motion. And there are three arms here with different diameter. So the way he makes the implants, let me show you the cartoon. They have different widths so that when they're placed around the nerve, they can circumferentially cover the whole nerve area. So you have a little locking tab here that goes through a hole, and this goes around the nerve, and like a Ziploc, you pull it, and it will capture that nerve as shown in this picture here. So you can present with this technology microelectrodes around the nerve and measure the traffic that goes through it. And what you see is that you can achieve sub-nerve resolution. For example, this electrode, that one, and that one, all measure different activity because, most likely because they're reading different fascicles. They're reading signals from their proximity and thereby are recording slightly different traffic. You can get a bit more insight into this by presenting electrodes along a nerve and recording the same event twice. So if you have a signal that goes from, let's say, muscle towards the brain, that signal will be recorded from one electrode and then another with a slight time delay. And by analyzing these time delays, you can start understanding where are those signals trafficked. For example, you can tell that there are different fibers inside these nerve fascicles. Each one has different diameter and transmits signals with a different velocity. So you can measure this in a qualitative fashion. And this is important because you can start latching on to these signals. For example, C fibers are known to convey signals that are interpreted by our brain as pain. And you can separate those signals out from all the other communication that takes place in the nerve. You can also stimulate very precisely. Here are stimulation data. Its point here corresponds to stimulation from a single electrode. And the letters here indicate the type of movement that's elicited. So this is the most articulate stimulation ever achieved from a peripheral nerve. By making microelectrodes and placing them around the nerve, you can get really exceptional a degree of motion separation. You can do better with implanted in the nerves, but that's invasive to the nerve. You can play the same tricks on the spinal cord. You can present, this is again the work of Ben Woodington, you can present electrodes on the circumference of the spinal cord, as shown here, and start measuring traffic in a very high resolution fashion. So here you can, for example, stimulate the sciatic nerve and cause sensory afferents that go through the spinal cord up on the brain. And you can capture those at the dorsal part of the spinal cord. So these signals, as they go towards the brain, they're trafficked at the back of the spinal cord. You can stimulate the brain and cause motor evoked potentials. These are motor commands that come down from the brain through the spinal cord to muscles to send them to motion. And those are manifested in a ventrolateral fashion. So they're in the front and side of the spinal cord. Once you map, then you know where to stimulate to achieve a particular motion. For example, by stimulating the ventrolateral part of the spinal cord, as shown here, and again, different points here correspond to different electrodes that are being stimulated to achieve different types of motion. And you can get even more complex movements using electrode combinations. Now, what can you do with this? You can start thinking of building bypass systems for spinal cord injury. So you can have two arrays above and below an injury site, and then capture a motor command as it comes down from the brain with this electrode array, and then electronically pass it to an array past the injury to stimulate the right location and elicit the intended motion. This is shown here. You can capture a motor command shown here, and then by closing this loop with a low latency interface, you can achieve muscle movement as seen by measurements of muscle activity and by kinematic analysis. If you disconnect this interface, then the motor command does not elicit any movement because of the injury. If you reconnect, then you bring the movement back. This is very crude at this point, but it points to a future where injuries of the spinal cord of peripheral nerves can be treated with this bypass technology. So we're very excited about this and now pursuing it in freely moving animals where we can get high quality recordings. We're already scaling up to porcine models. And the question we are trying to answer is how articulate can this stimulation be? To what degree can you bring back walking on someone who is paralyzed? Finally, let me tell you about the area that excites me the most, and this is the interface of bioelectronic medicine with regenerative medicine. What is regenerative medicine? We have today the ability to take cells from a human, bring them into a stem cell state, and then forward differentiate them to different somatic cell types. For example, you can make muscle, you can make neurons, and then transplant them back to the patient to repair damage caused by injury or disease. What if you were to combine these two technologies? So bring into the body not just tissue or implanted electrodes, but a combination of both. So we call this regenerative bioelectronics. And the idea is to combine electrodes with tissues, bring them into the body with the idea that the tissue doing some local repair through innervating the host or being innervated by the host, and then the electronics controlling this process. So here, the regenerative component, the tissue that you bring in, will camouflage the electronics from the body, increasing biocompatibility. It will repair the tissue, and then it will enhance communication between the electronics that you implanted and the host. At the same time, the bioelectronic component will monitor the process of repair. It could potentially even guide it, and then it will end up controlling repair tissue. So to show proof of principle, we went to a peripheral nerve bypass, injury bypass, which is much more difficult to do than a spinal cord one. The reason for that is the following. In a peripheral nerve, let me give you the clinical context first. If there is a peripheral nerve injury that's over 1 centimeter long, then you cannot connect back the two stumps, and people end up with a functional deficit. A way to bypass that functional deficit is to record motor commands as they come from the brain down to the proximal stump, and then take these commands to a computer that will interpret them and then use them to drive another implant that will convey that information to muscle. Unfortunately, when a nerve is injured, the axons here pull back. So the axons here at the distal stump are not connected to the cell body, which is in the spinal cord, so they wither away. And this stump will pull back. So this ends up being electrically dead conduit, and you have a hard time recording from it. What we try to do is to repair that conduit by giving it muscle tissue here. And the idea is as follows. You make an implant, and you culture on it muscle cells. So these come from stem cells that are forward differentiated into muscle. That takes place ex vivo, takes about 10 days. And then you would present the implant that contains electrodes and muscle onto the stump of the nerve. And here's the data that shows that this thing works. So here is the severed nerve. and we stimulate it with these two electrodes. If you present here just an electrode array and you measure from different electrodes, you measure only the stimulation artifact. There is no signal here, meaning that this part of the conduit is dead because the actions pulled back. However, if you present an implant that also has muscle cells on it and wait for a couple of weeks, then these actions will regrow. And then when you stimulate, you see this characteristic echo that tells you that there is signal conduction through these actions. So this also works in spontaneous activity recordings. You place the implant in. These are recordings week 1, 2, 3, and 4 from one electrode shown here. Week 1, week 2, you see very little activity. But after week 3 and 4, you start recording really beautiful muscle activity every time the animal moves. How do you know that? We correlate with behavior. And there is a correlation between the recordings that we capture and movement of the animal. So we know we are recording indeed motor commands. So this is very exciting because it shows that you can now repair the nervous system with stem cell tissues and then interface with it electrically. Now we're working on the distal part of the interface. We're presenting implants with lower motor neurons, hoping that they will innervate through the stump, the muscle, and myelinate in order to convey these motor commands that we record to the muscle. There are lots of questions. How do you best do this low latency link? What is the optimal resolution? Can we go to single axon? What happens in more complex tissues? And importantly, can we use electrical stimulation to enhance connectivity and speed up the repair process? So with that, let me summarize what I've discussed today. I hope I conveyed my excitement about bioelectronic medicine and how it represents a new way to treat disease by interfacing with the nervous system. Thin film implant technology, meaning that using the tools of microelectronics manufacturing to make implants in an automated process, is a game changer. It allows very thin implants to be made that can be activated for minimal invasive interfaces. It enables these ultra-thin cuff electrodes that go around peripheral nerves and spinal cord with new applications in injury bypass systems. And the interface with regenerative medicine paves way for new therapies. Now, let me take 2 slides and project into the future as to where this goes. We live in the era of what I call bioelectronic medicine version 1.0. All the devices I talked about today operate by stimulating tissue. And what that means is you either polarize or depolarize the membrane of neurons, and you get them to fire action potentials, or you inhibit them from firing action potentials. However, when you implant electrodes today, you implant them typically in an area that is compromised by disease or injury and can further degrade by neurodegeneration. So you're playing a game where you have to deal with the nervous system as is, often compromised and often getting more compromised with time. Can you do better than that? Turns out electric fields can do much more so than stimulate cells. There is a lot of literature that shows that electric fields can cause cells to migrate directionally towards one of the electrodes, can get cells to differentiate. These are immunochemistry, immunostaining patterns that show different states of cells grown under controlled conditions and grown under DC pulses. You can control cell adhesion, cell function, axonogenesis. You can coax neurons to grow new axons and reconnect with other cells. These phenomena, by and large, are very poorly explored and not understood, but they paint a picture of bioelectronic medicine 2.0, where you can use electricity not only to control the network as is, but to change it, to help, for example, regrow and repair the nervous system. How do we do that? We first need to understand the fundamental phenomena. The way I see this is as a phase diagram where you plot, for example, stimulation amplitude and stimulation frequency, and you want to map on this diagram the different phenomena, for example, migration, differentiation, axonogenesis, and find a set of parameters that you can use to elicit this phenomena. You do that in vitro and then eventually transition to in vivo, which is more complicated but eminently doable. So very bright future and a lot of work to be done. Let me thank the group. The group is co-led by a neurosurgeon and 2 neuroscientists. People on this list have contributed with backgrounds ranging from physics, chemistry, to engineering, to neuroscience, to neurosurgery. Collaborations in Cambridge and abroad and outside. Funding agencies is very important. Ideas without funding are mere hallucinations. So thank you for your attention. Thank you for the invitation. It's a pleasure to be here.
Well, I don't know about you, but I found that simply astounding. Do start thinking of some questions. We've got no questions online. If you are online, use the Q&A facility. I'd like to ask a quick, naive question. The initial video of the man with tremor, that was just remarkable. Utterly remarkable. Was that a one-off, or is that a reproducible result?
No, it is reproducible. It is an intervention that takes place for people with Parkinson's. There have been people implanted 20 years ago, and still there is some degree of efficacy maintained. It's a neurodegenerative condition, so eventually it loses efficacy, but typically you intervene with DBS after pharmaceuticals stop work or they elicit too many side effects.
Okay, thank you.
You've shown us how the dream would be, say, to have a severed nerve and to be able to control a hand or something. Would it be possible to have a simple mechanical hand, a robotic hand, that didn't have such range of movements?
Yes. So today, the state-of-the-art in myoelectric prosthesis uses electrodes on the surface of the skin, on the skin on the stump. Unfortunately, they're not very articulate, and often this is supplemented by introducing other control parameters. For example, you can have a little switch on the foot, and when you tap your foot, then the prosthesis could execute a particular motion. So there is a lot to be desired still. Remains a lot of work to be done to control such prosthesis and make them more articulate.
I've got an online question here from Paul Klein. Do you see mental health applications, perhaps schizophrenia or depression or migraine, which would be amazing?
Yeah, deep brain stimulation is being used to treat severe forms of depression that cannot be treated by drugs. It is prescribed today for that reason. The mechanisms are still, again, we do not understand the brain, still to be elucidated, but yes, this started happening in 2000, and it's, I think it's slowly taking off.
Thank you.
Hi, good evening. Another question related to mental health. I wonder, could you say a little bit more about the electrostimulation for OCD?
Yes.
What's the intention behind the stimulation, and what kind of results maybe are being seen so far?
Yes, I'm not an expert on that. I know that it is being used for extreme forms of obsessive compulsive disorder, but I don't know what the target is and what the mechanism is. I can point you to the right literature review.
Forgive me, I'm a little skeptical. Many years ago, I was party to electroconvulsive therapy for many diseases, through severe depression to schizophrenia and many other poorly labeled mental diseases. We found that 30% of people would claim that they had made benefit, which meant that they didn't have to have any more. 30% got worse and 30% the same, which I think was essentially tossing a coin.
Yeah.
How much validation of the success of your treatment are you going to be able to guarantee is a genuine success?
Yeah. So for conditions such as tremor, Parkinson's, it's very easy to latch onto a metric and show improvement, right? That's one component of the answer. The other is that with anything that is outside the brain, not implantable, the resolution is very poor. So you really don't know where you're stimulating. So there is transcranial direct current stimulation, and electroconvulsive therapy is an extreme form of that, where you, with 2 electrodes, induce a generalized seizure. The current that you inject with these electrodes goes all over the brain and causes what could be described as a major reset. What happens, I don't think anybody knows. We do not understand the brain enough to understand what happens after you recover from that seizure. People describe it as the brain being trapped to a local minimum, and by resetting it, you might help it go out of that minimum and to another state. But it's very crude, our understanding of the technique. Even worse, the metric for improvement is not very quantifiable. And I agree with you, electroconvulsive therapy is not well characterized in the benefit that it endows. NICE, which is the body that weighs in on use of technology, has authorized the use of, has given the thumbs up for the use of ECT by saying that it can be used basically as a last resort after everything else has failed. I think that sums it up, that we don't have, when nothing else works, you give that a try. It's not a characteristic intervention of bioelectronic medicine.
So I was interested by the idea of routing around nerve damage, and I understand the nerve, the electrodes that were connected to the upper nerve, if you like, being able to record what was coming from the brain.
Yeah.
What I didn't understand was how you do the mapping on the electrode on the severed part.
Yes. So what you can do is either map one for one or connect and let the brain figure it out. So the brain can easily remap. And the thing that I showed there is a little bit dated. Oh, hold on, where is it? Yeah, this one. So here, yes, you could map one electrode on the top array with one on the bottom and make sure that they represent the right location, or just randomly connect and the brain will figure it out. You can go a step beyond. You actually do not need a computer here with electronics. Muscle is a very good biological amplifier. So when you're recording some activity that comes down the nerve, the muscle activity is amplifying that signal. And all you need is a little bit of passive electronic circuitry to change the form of that excitation into something that can excite a neuron. So the devices can be much simpler than what is indicated there.
I have another online question from Andrew in Cambridge related to this. Once peripheral nerve stumps start to regrow with the help of muscle tissue, is there a possibility that eventually they will reattach and start functioning themselves again? I think it's similar to what you
Yes.
answered.
So the question is, if you were to take a bit of muscle, dangle it on a string, and keep pulling it, will the axons follow? I haven't seen anything done along these lines, but it's a very interesting question. How can you come up with a device that would perhaps take that muscle and start gently pulling it, and then bring it to the muscle you're trying to connect with? I don't know. Yeah.
Okay.
I was wondering how widely adopted this therapy is in clinical practice.
Which one? The
Any sort of deep brain stimulation.
So deep brain stimulation is currently used after drugs have stopped working or they elicit too many side effects. It's very hard to give a generic answer. For example, for pain, neuromodulation for chronic pain. Today, the first line of defense is run-of-the-mill painkillers. Then there are non-steroid anti-inflammatory drugs. If those don't work, the next step up would be either opioids or neuromodulation. A lot of people go for opioids instead of neuromodulation because the safety profile of neuromodulation is a little bit worse than drugs. There is a risk of infection, typically 0.5% or so. Again, depends on the intervention. People can be a bit squeamish about getting an electrode in their body. And it's much more expensive. It's a front-loaded cost to the system. And unfortunately, nationalized healthcare systems do not have a good mechanism for coping with that. In the case of Parkinson's, you would start with L-DOPA. When that stops working, the next treatment is DBS.
Got not so much a question as a comment from Peter Burning, but it might be of interest. I've heard of DBS for Parkinson's on a podcast, Movers and Shakers. Some of them have experienced it. So if you're interested, you might Google Movers and Shakers.
Cool. That's a good title for it.
Yes.
Yeah, thank you. I was interested in the implantable device and how you communicate with that, and also whether it's possible to have it rechargeable.
Yes, so there are implantable neuro stimulation devices. The actual, let me go back to the thing that I showed earlier. So this actually, I think the one that I show here is rechargeable.
By
Yes.
an induction.
Right. So this, if it has an R, it's rechargeable. And you bring a one to the body and you charge it. Patients don't like this. They'd much rather have a 10-year, I don't need to worry about this. And then in 10 years, I go get an incision, pull the thing out, put a new one in, than having to recharge it every month, every week or so. Yeah.
Really amazing talk and fascinating work. So I have a quick question, if you can basically expand more about the regeneration like cell therapy combined with the bioelectronics, because I was under the impression that the peripheral nervous system can regenerate, right? So if the axon breaks,
Yeah.
then it can grow back.
Yes.
So is that like to what extent is the bioelectronic side critical to this project, to this basically process? And then does it help with things like, remodeling of the nerve environment or something like that?
So in an injury of peripheral nerve, if it's below a centimeter, then you could bring the two stumps together, and then the nerve, the axons will regrow. But if it's larger than a centimeter, then you cannot do that. So that's, in this case, a bypass is a potential solution. Yeah,
So there's hope that the bypass can make it grow.
well,
back.
no, the bypass will make the, so the axon will grow inside the conduit and then connect with a device that you present on the conduit. It's not gonna bridge the gap. So what will be bridging the gap here is a cable. It's an electrical cable.
But is there hope for the axons to grow back?
There is a lot of work historically on trying to coax axons to regrow along the spinal cord during spinal cord injury. Not very successful. There was a lot of interest when Superman injured himself, and there was a lot of work trying to reconnect the spine electrically, but it hasn't yielded fruit. It's picking up again. There is a company called Onward Medical. This is run by folks out of EPFL, where they stimulate in order to elicit basically a reflex movement that simulates walking. So you might have seen it in the news the last 2-3 years or so. They got it into paralyzed people, and with the aid of a walker, they were able to walk under electrical stimulation. The mechanism is still debated, but it's most likely stimulating the reflex. That is, a lot of the walking that we do comes from the spinal cord. The brain engages only when you step into a pothole. So you can stimulate that reflex motion and get something that resembles walking.
Thank you.
We, about 3-4 years ago, we had a student award winner, I can't remember her name, unfortunately, from physiology, who was mapping by fluorescence microscopy the agents traveling along an axon, carrying repair materials to a break. I don't know how that, Marie, Maria Versalis, perhaps her name was, in physiology. I don't know how that work is coming on. So there are different avenues.
Yeah, yeah.
Another question on online from Mike Bailey. Given his concerns over the scattergun approach of ECT, do you have similar concerns around translingual neurostimulation, TLNS, for stroke rehabilitation, MS, TBI, etc.? I don't know. What, do you know what TBI
I'm not
is?
familiar with this acronym. I know that people stimulate to help with traumatic brain injury, but it's not mainstream. So electrical stimulation can have effects such as neuroprotection for reasons that are not understood. So it does make sense to explore electrical stimulation for helping with traumatic brain injury. Another thing you can do is during traumatic brain injury, you have a spreading depolarization that basically circles around the injured site of the brain. And every time that waves goes around, you have more and more damage in the area around the acutely damaged tissue. People try to stop that depolarization by applying electric fields, but I don't think that has yielded fruit yet.
Okay. Any further questions from the audience? I just have one question left from me. Okay. Mine's a physicist question.
Yeah.
What's the nature of the signal that is picked up and then reapplied to the nerve? Is it a spike? Is it a frequency, a sine wave, or?
So if the electrodes are very small, you can pick up spiking activity that comes either from action potentials coming down the axon, or from exocytosis events, or from the uptake of ions back into the cell.
But it's basically a sharp spike.
It can be a sharp spike if you record very close to the neuron with small electrodes.
Otherwise it gets...
Yeah, and if you make your electrode bigger, then you're recording from a larger neighborhood, and you measure the spatiotemporal superposition of everything. So what you measure with an electrode is the dance that little ions do right in front of your electrode.
Okay.
And they do this dance because they couple to every neuron in the neighborhood.
And what kind of time scale is that on? What's the rise time?
So the action potential is milliseconds. So you're looking at kilohertz, very slow activity.
Oh, and it's repeated. Ding, ding, ding.
There are typically the language of the brain is in frequency. So when a stimulus is applied and the amplitude is increased, a neuron would respond by increasing the frequency of spiking.
Thank you. Well, I thought that was pretty amazing. Thank you very much, George. That was an absolutely stunning talk.
Thank you very much.
We could get you back.
A great pleasure.
All of this work.
Thank you.
And thank you again.
Thanks a lot. Thank you.